Daniel Senahebo. I'm moderating for Miss Taylor Smith. She's a 26-year-old non-thesis student of the School of Agriculture and Environmental Sciences. She hails from New Orleans, Louisiana, where she received her Bachelor of Science degree from University of Louisiana State University in resource ecology and management with a concentration on wildlife ecology. She aspires to become a veterinary cardiologist if accepted into the veterinary school at the State University. She'll be presenting her research proposal this morning. May you all be welcome, Miss Taylor Smith. Thank you.
Let's go, Taylor.
Thank you, Dean, for that introduction. As Daniel just said, I am Taylor Smith, and today I will be talking to you about the evaluation of antioxidant enzymes SOD2, SOD3, and catalase in the developing Caprine testis. One of the fastest growing segments of livestock production in the United States is the caprine industry. However, the development of efficient goat production techniques are still in beginning stages. As goat meat is a new enterprise for American producers, the market infrastructure currently available to cattle, poultry, and swine producers have not yet been developed for goat producers. Assisted reproductive technologies, or ART, have the potential to rapidly improve goat production, but this requires a vast understanding of the molecular mechanisms associated with testicular function in regards to spermatogenesis. In an effort to meet the increasing demand for goat production, ART allows us to rapidly propagate superior genetics, giving us the opportunity to bring the goat industry forward more quickly. For some ART, germ cells are isolated from testes and cultured so that they can be enriched to increase numbers and can then be used for artificial insemination, transgenesis, and in vitro fertilization, and other technologies. All cells are abused by thousands of free radicals every minute of every day. A free radical is a molecule with an unpaired electron that is extremely reactive in an attempt to reach an electronically stable state. They are produced as byproducts of normal cellular metabolism, generated by chemicals in our environment, and are present in the air that we breathe and the food that we eat. In the testis, for example, developing germ cells, in addition to undergoing complex cellular changes, must migrate progressively from the basal compartment here to the adluminal compartment here, and are eventually released after the spermatids are elongated into the tubular lumen at spermiation. This process, per se, requires extensive tissue reconstructing in the seminiferous epithelium, which results in the production of reactive oxygen species. Reactive oxygen species include superoxide, hydroxyl, peroxyl, hydroperoxyl, hydrogen peroxide. If left unchecked, these radicals can induce oxidative stress, which results in cell damage and eventually death. This is just a visual representation of free radicals and oxidative stress damage to cells. So you have the normal cell here that is being attacked by free radicals or reactive oxygen species here, and it will undergo oxidative stress and eventually necrosis. And oxidative stress is basically just defined as the destruction or damage of cells or tissues by reactive oxygen species. So free radicals have the ability to directly damage sperm DNA by attacking the purine and pyrimidine bases. ROS can cause damage via single and double-stranded DNA breaks, crosslinks, and chromosomal rearrangement. ROS-induced damage of mitochondrial DNA leads to decreased ATP and energy availability, which impedes sperm motility. And decreased motility has also been associated with ROS-induced, primarily hydrogen peroxide-mediated peroxidation of lipids in the sperm membrane. To maintain physiological oxidative mechanisms while minimizing the risk of cellular injury, a balance between ROS and antioxidants is necessary. Antioxidants are reducing agents that react with and buffer reactive oxygen species and are considered a form of defense against oxidative stress. Important enzymatic antioxidants involved in spermatic function include superoxide dismutase, SOD, and catalase, CAT. So this is just a visual representation of the enzymatic antioxidants that react with and buffer free radicals. As you can see here, you have the free radical superoxide. It is dismutated by SOD into hydrogen peroxide here and into molecular oxygen here. And then catalase will come in and convert this hydrogen peroxide into molecular oxygen and water, thereby relieving the cell from oxidative stress. SODs are metalloproteins, so depending on the transitional metal ion found at their active site, SODs can be categorized into one of three types. Iron SOD or SOD1, which is found in prokaryotes. Manganese SOD or SOD2, which is found in the mitochondria of eukaryotes and prokaryotes. And copper zinc SOD or SOD3, which is localized predominantly in the extracellular matrix. For the purpose of this study, we will focus on SOD2 and SOD3. Catalase is a heme protein that is located predominantly in peroxisomes and in the inner mitochondrial membrane. Increasing evidence suggests that catalase may be an important enzyme in the metabolism of lipids, especially cholesterol. Catalase-bearing peroxisomes have been described in the Leydig cells of the rodent testis and in the Sertoli cells of the rabbit testis. And so based on that fact from our literature review, we hypothesized that SOD and catalase will play a role in testicular function in goats as well as the other mammals. The objective of this study is to determine the temporal and spatial levels of the antioxidant enzymes SOD2, SOD3, and catalase in the testes of neonatal, prepubertal, and adult goats using immunohistochemistry and Western blot. And then we'll correlate the findings with the morphological changes that occur during testicular development. We will be using tissues that have already been harvested from pico-boar-crossed goats at 3 different life stages. The neonatal group will be castrated between days 14 and 28 of age. The prepubertal group will be castrated between days 60 and 120. And then the adult group will be castrated when they are greater than 180 days of age. And we will be sampling or using samples from goats in 3 different goats in each of these life stages. After preparing the paraffin-embedded tissue and the protein lysate from the samples, they'll be stored at -80 degrees Celsius until further analysis, and then densitometry will be used to quantify the protein levels. So the first protocol that we'll be using is immunohistochemistry. So paraffin-infiltrated tissue samples will be sliced using a microtome and then mounted onto charged slides. The sections are then deparaffinized and rehydrated before antigen unmasking is performed. They are then exposed to a primary and secondary antibody, which binds to the target protein before chromogenic staining. And once the secondary antibody has a specific enzyme that's attached to it, so once the corresponding substrate is added to the samples, a color will be emitted. The other procedure that we'll be using is Western blot. So we will take tissue samples and create a protein lysate from them. We will add the samples to the gel in between the gel cassette and then put it in accordingly, add the running solution, start the electrophoresis, and then the proteins will be separated based on their weight onto the gel. From the gel, the proteins are transferred onto a membrane and then exposed to a primary antibody followed by a secondary antibody. So here is the protein that we're targeting. We add the primary antibody, which is here, the secondary antibody, which binds to the primary. Attached to the secondary antibody is an enzyme, and so once we add that substrate, a color will be produced. proteins are going to be visualized using the chemiluminescence, which will qualify the amount of protein per sample. The thicker the band, the more protein in that sample. So it is expected that immunohistochemistry will show that SOD2, SOD3, and catalase will be present in all life stages, but the levels will be greater in neonatal testes as compared to that of the prepubertal testes, since spermatogenesis begins to occur in the prepubertal life stage. It is expected that Western blot analysis will demonstrate a significant decrease in enzyme levels in the adult testes as compared to that of the prepubertal and neonatal. To better understand, the expected impact of the research is to better understand the molecular mechanisms of spermatic function, which may lead to enhancing existing or developing new assisted reproductive technologies. And this research will be a beneficial addition to the oxidative stress index in ruminant and not only veterinary medicine, but medicine worldwide. I would just like to acknowledge my advisor, Dr. Olga Bolden-Tiller, and my committee members, Dr. McElhenney and Dr. O'Keery. I would also like to give a special thank you to Dr. Calda, as well as Jamia Mason. Funding for the research was provided by the USDA through TUSEP and Evans Allen. Thank y'all for listening.
Thank you. May I open the floor for first the registered students, graduate students, the non-registered before the faculty? Yes.
Julie Mitchell, does the breed of goat, do you think, affect the results? Do you think if you use a different type of breed, will it have different results, or is it just the ages?
I think it's just going to be the ages that'll make a difference in the results. There are antioxidants in all animals, so I'm sure that you would find the same levels in different breeds of goats.
Shaniqua Bynum, can you explain what free radical does?
Yes, so I'll go back. So a free radical is a molecule that has an unpaired electron in its outer shell, and so it's extremely unstable and tries to react with all major classes of biomolecules in an attempt to become electronically stable.
So you're saying how
Could you state your name?
Oh, Rashad Brown, you were saying how the oxidative stress kind of affects like the DNA within the sperm cell. So is this like known to cause sterility within the testes?
Yes, reactive oxygen species have been found to be a major cause of infertility in the male.
Aaron Gary, you stated that you're using the enzymes SOD2 and SOD3. Why aren't you using SOD1?
I am not using SOD1 simply because SOD1 is only found in prokaryotes, whereas SOD2 and SOD3 are found in eukaryotes.
This will be the last person, then we'll move to the faculty.
Dorian McFarley, is there a certain reason why you use the tissue samples from the Kiko and Bora goat?
As opposed to other breeds of goats. I think that those are just the goats that we, have got the samples from previously. I'm not sure specifically why we use the Kiko and Bora goats. I know that those are what's available to us here at Tuskegee. So,
May I use this opportunity to invite the questions from the faculty?
What about non-registered?
Non-registered? Is there any non-registered student here who has a question? Okay. Let's get it. Okay.
Miss Taylor, so you talk about two methods you're going to Western and the ELISA technique, I think.
Immunohistochemistry, yes, ma'am.
Yes. Okay, so the immunochemistry will be in tissue or in ELISA.
It'll be in tissue samples.
tissue.
Yes, ma'am. So the tissues, like once we actually harvest the testes, we'll put them in formalin to preserve them, and then we'll cut them into thin slices, and then mount those slices onto a microscope slide so that we can observe them that way, microscopically.
Okay, so what technique are you utilizing for your protein extraction for your Western?
For the Western blot, the proteins will be homogenized. I haven't actually prepared the protein lysate yet, so I'm not exactly sure of the methodology, but I do know that they will be homogenized, and all proteins that are available in the cell will be available in that lysate, as opposed to just my specific target protein.